Stop what you're doing. Look at your hand.
If you cut it, just a little, in the early 1900s, you were facing a coin flip for your life. Infectious diseases were the unseen executioner, making life brutally short. The average person lived to a meager 47 years old. Pneumonia, blood poisoning, tuberculosis, these weren't illnesses, they were death sentences. The early years of the 20th century were defined by a silent but omnipresent terror, the unseen killer that lurked in every cut, every scratch, and every breath. Infectious diseases constituted the overwhelming majority of human suffering and mortality worldwide. Life, even in the industrialized capitals of Europe and America, was brutally short. The average life expectancy at birth hovered at a meager 47 years. Diseases like pneumonia, septicemia, blood poisoning, diphtheria, typhoid fever, and tuberculosis swept through populations without discrimination, defining the leading causes of death. To contract a serious bacterial infection was often a guaranteed, painful sentence. Facing this overwhelming microbial threat, medical practitioners were largely powerless, often resorting to remedies rooted in antiquity. For over 2 millennia, bloodletting remained a standard, universally accepted treatment for almost any ailment, including virulent infectious diseases. Physicians, in a futile attempt to alleviate symptoms, withdrew blood using crude instruments like syringes, lancets, and even leeches. These practices were not only ineffective but frequently worsened the patient's condition, accelerating the decline rather than slowing it. The desperate state of medicine revealed a critical conceptual flaw. There existed no reliable method to kill the invading microbes within the body without simultaneously poisoning the patient. Then, in 1910, came the first crack of hope, the magic bullet. Paul Ehrlich developed a drug, salvarsan, that could specifically target one terrible infection, syphilis. He proved it was possible to kill a germ without killing the patient. The introduction of salvarsan, followed by its derivative neosalvarsan in 1912, marked the dawn of chemotherapy, offering the radical concept that one chemical could selectively target a pathogen. For 2 decades, these arsenic-based compounds remained the sole reliable chemical agents for treating bacterial infections, ushering in a complete transformation of syphilis therapy. But the magic bullet was toxic, difficult to use, and couldn't touch the common killers like sepsis. Salvarsan was medically difficult to administer, partly due to solubility issues, and was decidedly unsafe for the gravely ill. While Ehrlich achieved a monumental milestone, he fell short of his own assigned goal, a single injection cure, and his success was not readily replicated for the more common, acute infections like sepsis or pneumonia. The toxicity hurdle remained the central paralysis of medical science. How could one find an agent potent enough to kill robust bacteria, yet benign enough to leave human cells unharmed? A brief surge of optimism came in the 1930s with the development of sulfonamide drugs. The German biochemist Gerhard Domach introduced Prontosil, which found limited but definite success in treating bacterial infections. French scientists soon clarified the paradox of Prontosil's success in vivo, in the body, but failure in vitro, in the test tube, determining that the active compound was sulfanilamide, metabolized from Prontosil within the patient. The success of these derivative sulfa drugs changed the pervasive cynicism surrounding bacterial chemotherapy, demonstrating that chemical solutions for general infections were indeed possible. They reigned supreme in the fight against bacteria until they were ultimately displaced by the true revolution that had already been briefly observed, but overlooked, a decade earlier. The foundation of medical thought was finally shifting from medieval practices toward molecular targeting, yet the perfect, non-toxic weapon remained elusive. Chapter 2: The Temperament of St. Mary's. The stage for the true revolution was set not in a grand industrial laboratory or a state-of-the-art chemical facility, but in the cluttered, third-floor laboratory of St. Mary's Hospital in London. There, the Scottish physician and bacteriologist, Alexander Fleming, pursued his quiet, diligent work. Fleming was no stranger to the search for the body's natural defenses. Years before, in 1921 to 1922, he had already stumbled upon an enzyme in human tears and nasal mucus that could cause bacteria to disintegrate. He named this enzyme lysozyme. This prior experience was fundamentally important, serving as an advanced sensitizer for Fleming's eye. Lysozyme, while demonstrating natural antibacterial action, proved largely ineffective against the dangerous, disease-causing pathogens that stalked his patients. Furthermore, all attempts to concentrate the enzyme to strengthen its antiseptic properties failed. Fleming understood acutely the difficulty of harnessing a natural, non-toxic biological agent that possessed the necessary potency. This historical context meant that when he encountered another instance of natural antagonism between life forms, he was uniquely prepared to appreciate its significance. The epical moment arrived in September 1928. Fleming, while investigating the varied growth of Staphylococcus aureus cultures, returned to his laboratory following a summer vacation. He was met not with sterility, but with an unexpected sight. Some of his Petri dishes had been contaminated by a common airborne mold. Had he been a tidier, less inquisitive scientist, he likely would have summarily discarded the dishes, and the trajectory of human history might have been vastly different. Instead, he placed the contaminated dish under his microscope. What Fleming observed was a stunning biological phenomenon, a clear indicator of the invisible war perpetually waged in the natural world. Surrounding the invading mold, he saw a pristine, clear zone, a halo of death where the robust Staphylococci bacteria could not grow. This zone was proof that the mold was actively manufacturing and secreting an agent lethal to bacteria. The antibacterial power demonstrated by this mold was far superior to the gentle effects of his earlier discovery, lysozyme. Fleming quickly cultivated the mold, which he identified as a variant of Penicillium, later confirmed as P. chrysogenum, now P. rubens. He concluded that it produced an antibacterial agent that not only inhibited growth but could potentially combat serious infectious diseases in humans. He christened the active substance penicillin. Looking back on the moment of discovery, Fleming later reflected with characteristic modesty and drama. "When I woke up just after dawn on September 28, 1928, I certainly didn't plan to revolutionize all medicine by discovering the world's first antibiotic or bacteria killer. But I guess that was exactly what I did." Chapter 3. An Observation, Not a Cure. Following his initial observation, Fleming dedicated the remainder of 1928 and 1929 to documenting the substance's properties. He successfully collected data that unequivocally established the chemotherapeutic potential of penicillin, focusing particularly on its effect on Staphylococci and other gram-positive pathogens. Crucially, his early tests quickly separated penicillin from the toxic legacy of salvarsan. Fleming observed that an extract of the mold broth showed no harmful effects when injected into the ear of a rabbit, nor did it cause any adverse reaction when applied directly to a human wound. This non-toxicity was the truly revolutionary aspect, proving that the magic bullet could indeed be safe. He formally published his seminal findings in 1929. However, the journey from laboratory observation to clinical application proved to be an insurmountable hurdle for Fleming and his team. While he possessed the microbiological understanding, the subsequent challenge was one of chemistry and chemical engineering. Penicillin, as produced in the laboratory broth, was a highly unstable compound. Every attempt by Fleming to isolate, purify, and concentrate the active substance from the raw mold juice resulted in the compound breaking down, frustrating his efforts. The purification process, requiring specialized techniques to handle a molecule that was sensitive to temperature and pH shifts, was simply beyond his capabilities as a bacteriologist. The significance of the discovery was therefore not fully recognized by the wider medical community. Paul Ehrlich's vision of the magic bullet, while proven possible, seemed just as elusive as ever, and interest in pursuing complex natural antimicrobials waned in the early 1930s. Fleming, disheartened by the chemical instability, effectively gave up research on it by 1932. Yet, he remained convinced of its potential, sending cultures of his Penicillium mold to anyone who requested it, nursing the hope that another scientist, perhaps with a background in chemistry, might succeed where he had failed. The decade that followed was a period of therapeutic dormancy for penicillin. Its biological power was known, but the technical gulf between culture broth and stable, usable drug was too wide for the prevailing specialized science to bridge. The world waited, unknowingly, for the right combination of expertise, drive, and geopolitical necessity to converge and resurrect the unstable magic found on a simple contaminated dish. The true success of penicillin would not be defined by the initial lucky observation, but by the systematic, relentless labor required to overcome its inherent chemical fragility and turn a laboratory curiosity into a marketable, life-saving pharmaceutical. The Oxford Crucible and the Race Against Time, 1939 to 1941. Chapter 4. The Pathologist and the Refugee Chemist. A full decade after Alexander Fleming's publication, the fragile hope of penicillin lay forgotten, a mere footnote in bacteriology textbooks. The outbreak of World War II in 1939, however, created a renewed, desperate urgency to find agents capable of treating battlefield infections, which historically claimed far more lives than combat itself. The critical resurgence of penicillin research began at the Sir William Dunn School of Pathology at the University of Oxford, an institution newly equipped and led by the Australian pathologist, Sir Howard Florey. Florey, a dedicated researcher known for his scientific drive, had arrived at Oxford in 1935. In 1939, he shifted his departmental focus to investigate antibacterial substances systematically, specifically turning his attention to Fleming's work. The project became a necessity as Britain braced for War.8. To conquer the chemical barriers that had defeated Fleming, Florey assembled a formidable and complementary team of specialists. Central to this group was Ernst Chain, a German-born Jewish biochemist who had fled Nazi Germany. Chain was a man of intense scientific rigor, and his selection of penicillin for study was, ironically, driven by the profound chemical challenge posed by its notorious instability. Chain undertook the initial and most daunting task, extracting and identifying the active principle from the mold culture. The convergence of Florey's decisive leadership and Chain's meticulous biochemical skill was paramount. Chain immediately confirmed the strong antibacterial activity of the mold filtrate, but struggled with the compound's extreme sensitivity. Penicillin would rapidly decompose when isolated, confirming Fleming's earlier failure. The team soon realized that producing enough material for systematic animal testing, let alone human trials, required an industrial-level effort. They needed to process up to 500 liters of mold filtrate weekly just to yield a minuscule amount of usable compound. The solution required not just chemistry, but ingenious technical engineering. Chapter 5. The Architect of Extraction. The unsung hero who transformed penicillin from a chemically fleeting observation into a therapeutic reality was Norman Heatley, a young biochemist on Florey's team with an innate genius for invention. Heatley was aptly described as the key technical man. Where Fleming lacked the chemical means to isolate the drug, Heatley provided the necessary engineering toolkit. Heatley's contributions were twofold and fundamentally critical to the project's progression. First, he devised a reliable method to measure the potency of any sample of penicillin, the cylinder plate diffusion technique, which later became known internationally in Oxford units. This assay was essential because it allowed the team to track whether their various experimental purification steps were actually yielding active drug or merely inert impurities. Second, and most critically, Heatley invented a process that finally solved the long-standing problem of chemical instability. Utilizing a multi-stage countercurrent extraction process, Heatley built a semi-automatic apparatus from repurposed glassware and various bits and pieces. This ingenious system exploited penicillin's solubility characteristics. It allowed the penicillin, which was initially in an acidic aqueous solution, to be quickly extracted into an organic solvent, amyl acetate, and then immediately back-extracted into water by neutralizing the pH. This rapid, continuous cycling prevented the compound from decomposing, which it would do if left in the unstable acidic phase for too long. The result was the first usable, albeit crude, powdered form of penicillin. Because conventional glassware and industrial equipment were scarce under wartime conditions, the resourceful Oxford team used a bizarre assortment of containers for growing the massive volumes of mold, including hospital bedpans, food tins, and milk churns. Heatley personally designed specialized ceramic culture vessels for optimal surface growth. The initial purified yield was shockingly low. The resulting yellow powder was only about 1% pure, but it was stable enough for clinical testing, proving that the technical solution to the drug's inherent fragility had been found. The ability of the Oxford team to move past Fleming's roadblock was achieved through the deliberate application of specialized biochemical engineering, confirming that the initial failure was one of scientific specialization, not lack of vision. Chapter 6. The Ultimate Test, mouse and man. With the first stable samples of penicillin powder available, the Oxford team proceeded immediately to the critical phase of in vivo testing. On May 25, 1940, a pivotal experiment demonstrated the drug's potential beyond the Petri dish. 8 mice were deliberately infected with lethal doses of virulent streptococci bacteria. 4 were left untreated as controls, while the other 4 received injections of the purified penicillin. By the next morning, the 4 untreated mice were dead. Yet, those that had received penicillin survived for days or weeks, proving definitively that the drug was not only non-toxic but overwhelmingly effective inside a living organism. The success of the mouse trials provided the moral and scientific justification for the first human trials, which took place under the shadow of the ongoing war. On February 12, 1941, Albert Alexander, a 43-year-old police constable, became the first patient treated with the Oxford penicillin. Alexander was suffering from a catastrophic, life-threatening septicemia, which had resulted in massive abscesses affecting his face, eyes, and lungs. His infection had reportedly stemmed from a small cut sustained while he was maintaining order during the Southampton Blitz bombing raids in November 1940, clarifying the popular, though incorrect, legend of the rosebush scratch. The results were nothing short of miraculous. After 5 days of continuous penicillin injections, Alexander showed a remarkable recovery. His fever dropped, his infection receded, and the deadly abscesses began to heal. However, the victory was tragically short-lived. The supply chain, relying on the inefficient surface culture methods and Heatley's painstakingly slow extraction apparatus, simply could not keep pace with the massive dose requirements needed to fully clear a systemic infection in an adult human. The team, in a desperate attempt to conserve the precious drug, even resorted to extracting residual penicillin from Alexander's urine to be purified and reused. It was not enough. The stock ran out, the infection relapsed, and Alexander died a month later. The tragedy of Albert Alexander was perhaps the most powerful catalyst for the drug's eventual mass production. His initial, spectacular recovery proved that penicillin was a genuine miracle cure, but his ultimate death proved that the medical community was utterly paralyzed by a crisis of scale. The drug was unusable unless it could be produced industrially. Recognizing the extreme risk posed by the German war machine to their precious research, Florey, Heatley, and other collaborators took an extraordinary precaution. They rubbed penicillium spores into the lining of their laboratory coats. The measure, immortalized in the phrase, "the mold in Dr. Florey's coat," ensured that if the laboratory were bombed or if Britain were successfully invaded, the essential fungal culture could be carried, hidden on their persons, and the life-saving research could be resurrected elsewhere. This necessity for global protection immediately preceded the mission to America. Tanks, Tycoons, and the Tide of War, 1941 to 1945 and Legacy. Chapter 7. Seeking Salvation in the New World. By 1941, the medical potential of penicillin was unquestionable, but the industrial reality was bleak. Britain, already ravaged by the Blitz and focusing its industrial capacity on traditional armaments, could not divert the resources necessary to scale production. Howard Florey and Norman Heatley recognized that the only solution lay across the Atlantic. In 1941, they traveled to the United States to enlist the aid of the American government and the powerful, burgeoning pharmaceutical industry. Their goal was clear: to move penicillin from the tedious academic surface culture method, which yielded barely enough for one patient, to a genuine industrial process. The timing was providentially strategic. Although the United States had not yet fully entered World War II, the military importance of a drug that could eliminate septicemia, the leading killer of wounded soldiers, was immediately recognized. The U.S. government initiated a vast, coordinated, government-sponsored project, forging an unprecedented coalition between academic researchers, the British team, governmental laboratories, like the USDA's Northern Regional Research Laboratory, NRRL, and major American pharmaceutical firms. The existing production method, surface culture, growing the mold on the surface of a shallow liquid medium, was fundamentally inefficient for industrial scale. To achieve the necessary output, researchers needed to adopt submerged culture fermentation, where the mold would be grown not on the surface, but throughout a vast, constantly agitated, and aerated liquid medium in deep tanks. This technical pivot required 2 parallel breakthroughs: a much higher-yielding strain of Penicillium, and the refinement of the fermentation engineering itself. Chapter 8: The Moldy Cantaloupe and the Deep Tank. The first breakthrough came from the Northern Regional Research Laboratory in Peoria, Illinois. A global search was initiated to screen various Penicillium strains, with soil samples shipped from around the world. Against all odds, the single most productive strain did not come from some exotic locale. It was discovered on a moldy cantaloupe found in a local Peoria fruit market. This strain, Penicillium chrysogenum, NRRL 1951, proved significantly more prolific in submerged culture than the original strain discovered by Fleming. Scientists at the Carnegie Institution and the University of Wisconsin further optimized this strain using X-rays and ultraviolet radiation, creating derivative strains that serve as the ancestors of all modern biotechnical penicillin production. The second, equally transformative breakthrough was the refinement of the deep tank fermentation process. This development was hailed as the critical development for making the drug viable. The methodology involved growing the aerobic mold cells in massive stirred tank fermenters, bubbling air through the liquid and agitating it with electric stirrers to stimulate enormous quantities of mold growth while maintaining sterility. The perfection and expanding uses of the stirred tank fermenter established an engineering pillar for the entire modern age of biotechnology. American pharmaceutical giants rapidly scaled up this process. Pfizer Incorporated, headquartered in Brooklyn, New York, made a pivotal contribution. Utilizing its expertise in fermentation, the company rebuilt an old ice plant, which already contained the essential refrigeration machinery required for the temperature-sensitive process, and opened the world's first large-scale deep tank fermentation facility in March 1944. The increase in supply was dramatic, requiring 24/7 operation. By the end of 1942, enough penicillin was available to treat fewer than 100 patients in the U.S. But by September 1943, supply was sufficient to meet the critical demands of the Allied armed forces. This triumph of chemistry and engineering provided a decisive tactical advantage. In preparation for the D-Day landings in June 1944, scientists worked around the clock to manufacture 2.3 million doses, securing penicillin's place as a strategic wartime asset. Chapter 9: The Apotheosis and the Aftermath. Penicillin's role in World War II was transformative. Coupled with advances in whole blood transfusion, the drug fundamentally altered military medicine. U.S. military surgeons during the Italian campaign and Operation Overlord credited penicillin with saving thousands of lives, dramatically improving wound treatment outcomes. Official medical history recognized that the control over sepsis represented the most striking contrast between the Normandy campaign and previous wars. For the first time, soldiers wounded in battle stood a strong chance of surviving infections that had historically been death sentences. The colossal effects of penicillin led to the awarding of the 1945 Nobel Prize in Physiology or Medicine, shared by Alexander Fleming, Howard Florey, and Ernst Chain. This recognized achievement cemented the trio's place in history as the men who ushered in the antibiotic era, transforming life expectancy globally and shifting the leading causes of death in developed nations from communicable diseases to non-communicable ones. However, the award generated significant controversy, primarily due to the conspicuous exclusion of Norman Heatley. Despite his contributions being undeniably critical, devising the assay, inventing the unique extraction apparatus, and providing the technical bridge between academic science and therapeutic application, Heatley did not share the prize. Historians and colleagues, including Florey's successor, acknowledged Heatley's critical role, encapsulated by the phrase, "Without Heatley, no penicillin." Heatley, though remaining modest, was later uniquely recognized by Oxford University for his essential work. The success of penicillin laid the foundation for drug discovery, transforming the pharmaceutical industry and leading to the search for similar microbial agents, which were collectively termed antibiotics. The ensuing decade saw a furious race for discovery, with the period between the 1950s and 1970s becoming known as the golden era of antibiotic development, yielding new classes of life-saving compounds like streptomycin, chloramphenicol, and vancomycin. Yet, the victory was immediately met by an emerging threat. Penicillin's legacy, while revolutionary, is ultimately defined by the initiation of a continuous, co-evolutionary struggle with bacteria. Fleming himself had worried about the misuse of the drug, predicting the bacterial counterresponse. His concerns materialized shockingly quickly. As early as 1942, before mass civilian availability, for distinct strains of penicillin-resistant Staphylococcus aureus were already documented in hospital patients. This rapid biological adaptation confirmed that the magic bullet, while potent, was only a temporary advantage in the arms race against nature. Decades later, with the emergence of multi-drug-resistant strains and the lack of novel drug classes since the 1980s, the world now faces warnings from organizations like the WHO about a potential post-antibiotic era, bearing chilling similarities to the bleak medical reality that existed before 1928. The discovery of penicillin, therefore, was not the end of infectious disease, but the decisive opening volley in a perpetual biological war.